So again, as we've done in previous sessions, we're going to spend some time now with these individuals that you've heard from, and we don't need introductions now. We've been introduced to each of our participants in this discussion section earlier in the meeting. But we're really going to be thinking about what are the elements that we would be needing to begin to map a strategy around research for the next decade. Again, in the context that we're looking at the interaction between microbiome and environmental health, and we're thinking about this from the standpoint of not only the basic science, but the application of that science to risk assessment and decision making. So how do we begin to think about this from the standpoint of the discussions that we've heard over the last day and a half, and mindful of the kind of progress that we've made in this field, as Lita has just talked about. You know, I think that there have been a number of suggestions made earlier on about the focus of function of the microbiome as a particular area of emphasis as we move forward, and I want to hear a little bit more about that as we get into our discussion. There's been some specific discussions. Matt, I think, talked about maybe looking for some biomarkers that would help us to characterize certain microbiomes, if you want to look at it that way, that would be important for us as we begin to think about the interaction with xenobiotics or with our environment more generally. So that was something that we've talked about. As much as we'd like to think about personalized medicine and precision medicine, that's not as useful to us in terms of risk assessment and regulatory decision making from an environmental health standpoint. So what is it that we can take from those kinds of activities that might bring us to the population or the communities that we need to begin to think about? So I'm going to throw a couple of those suggestions out on the table, and we just heard from Lita, but let me start down at the end there, Yvonne, and just get a couple of thoughts from you about either what I've just talked about or some other things that might be on your mind.
No, I definitely would like to echo what you just said, Bill. And I think that this is one of the classic situations where I think it's a rediscovered new field, and that is fueled by new technology and new interest from NIH. And I think many of us realize that the development in science is cyclical, and we're living through the new cycle of microbiology reincarnated as the microbiome, which is absolutely fantastic. And also very similar to other advances in the field, the medicine and the clinical needs and the human health applications are really moving first into this new area. And I'm very excited to not only have heard yesterday's, big human studies that collect multidimensional data sets, but also what Lita just described this morning. These are tremendously interesting data sets. And I think what the environmental health community, both the research and the regulatory communities, really has a lot to learn about, what works. And I take this in a sense of what data sets, what types of information are most informative. And I think, again, it's probably not very smart and maybe futile from the fiscal side of things to, again, start, building or think about large human cohorts or animal studies that would match this different dimensionality with environmental exposures, with disease component, if we try to collect everything. So I think I would encourage, HMP and other consortia to quickly come to grips about what are the most interesting and informative and not redundant types of information that are being collected. And as Lita mentioned at the very last exchange, what material is bankable and is actually amenable to longitudinal study designs, because we do want to do chronic, follow-up, and we do want to do chronic animal studies as well. So I hope HMP includes genetics as one of the data sets. It wasn't on the slide. I hope you're genotyping these individuals as well, because again, as were discussing yesterday, the inter-individual variability, everything is different about each individual, but we really need to understand, whether we're collecting, the same distribution over and over. And for that matter, these bioinformatic analyses, the putting all this information together are critical. And I wouldn't be averse at recommending that the environmental field, takes a year to wait and see what actually comes out of these before we start designing really our own studies, because our studies are even more complex. There are exposures that are layering on top of that, and that's something that will bring additional complexity, which I frankly don't think we can afford to just go everything against everything plus the exposures. And that's my personal feeling out of this day and a half.
So, Margaret, you've had an opportunity to look at a particular environmental contaminant in dealing with some of the metals and other things over your career. It seems to me that where we do have some opportunities to explore the microbiome along with some of these specific exposures, and again, with a focus on what are the functions of the microbiome that influence dose or response from these toxicants, that becomes a fruitful way to begin to look at this. And again, going back to something that Lita said, we really do want to understand, even though there may be a large diversity of genetics associated with these microbiomes, is there a real diversity of function of the microbiomes that we're looking at? And if the functions are more consistent, then the application will be easier to deal with. Margaret, do you want to just make some comments?
Well, I'll echo what others have said about this being a very exciting area of pursuit. And I liked what Ian said yesterday about the forgotten organ. I mean, this is a tremendous opportunity for us to understand health and the developing health and prevention opportunities in turn. And then building on, again, what Lita and Ivan said about collection. So we're conducting epidemiologic studies, and we have an opportunity to build on those studies should we collect samples appropriately, put them in RNA later. What is the freezing protocol? And I know the Human Microbiome Project has been very good about presenting protocols publicly. I think the epidemiology community does need to understand, if they are conducting a study, what are the right methodologies for collection and processing of those samples so that they can have fruitful data, so they can do metabolomics, transcriptomics, et cetera, as well as using the DNA. So I think the epidemiology community would like to see that maybe more, I don't know if it's like a consensus paper or, and maybe the epidemiology literature, so that as epidemiologists have these opportunities within these cohorts that are so expensive to assemble, where we try, even if we have a specific hypothesis in mind, we're going to look at multiple exposures and multiple outcomes because it's such a large undertaking and expensive. And then as we plan new ongoing accrual, what would we ultimately do thinking ahead? I mean, we're always needing to think ahead if we want these prospective studies of what might our samples be useful for in the future. We usually never know. We started collecting DNA before we knew about the SNP.
Thanks. It seems to me that, one of the things that has come out as what we should do next, which is this question that we're trying to address here, is this sort of recognition of the importance of the microbiome as Ellen Silbergeld has said, as a gatekeeper, a modulator that needs to be taken into account. And so the question of whether in our animal studies or in our epidemiologic studies we should be at the very least banking samples, I think is one that has received a lot of support at this point, and maybe something that as you just said, Margaret, is something that we should be thinking about without knowing exactly what questions we're going to ask of it. doing what we can to try to put those away to look at in the future, if possible. I think, Matt, in your presentation, you started to talk about the characteristics of the microbiome and the fact that we can begin to think about how those might even be modulated, and of course, looking at it from the drug standpoint, or in terms of efficiency of dosing, or looking at it from the standpoint of reduction in toxicity and so on. There are great parallels for environmental health as you begin to look at the possibility of perhaps modulating the gut microbiome in terms of environmental exposures that might in fact reduce the amount of chemical that's getting into the body. Have you thought more about those kinds of things given this meeting in terms of the environmental health perspective?
Yeah, I definitely have. But before I talk about that, I just want to make a case for something that I haven't heard yet that I think is particularly crucial, which is the effects of environmental toxins on plant-associated microbes. So these are compounds that are being leaked out into the biosphere all the time. And the USDA is here, and I know they're participants in a lot of this, but that's an area where, and maybe that's associated with the kind of environmental or habitat sampling. But I feel like that's really crucial. That's something that we know the roots have very intimately associated microbiomes. They're as essential to the roots as they are to us. The root enclosures that contain those microbiota are very much like our intestines. And to my knowledge, nobody has really delved into how environmental toxins might affect that. And I know this is a health decisions type meeting, and this is a little bit expansive from that, but I do think that's something we want to put on the table and have be a consideration. Again, the technologies exist. I know there's somebody at Carolina that has already done a lot of root microbiome work, so it isn't, you wouldn't have to reinvent the process. So I think I'll just leave my comment at that at this point. I said a lot of things in my presentation about where I thought human health and toxins and microbiota modulation could go, and I feel very confident that we will get there and have an effect.
And I think that is it's obviously a very important point for our ecotoxicology colleagues who are interested in the questions of these kinds of changes that might have impacts from an ecological standpoint. But Lita also talked about the fact that, we interact with our environment, and the changes in these various microbiomes do have some impacts on humans, because that's where we're getting our repopulation and changes in those populations from. And maybe, Lita, do you want to say anything about Matt's comments?
I do actually want to make a couple of comments. I hope they address some of Matt's comments. I want to note that the USDA is apparently starting to mount a phytobiome initiative. So I met with some Isabel, I met with some USDA colleagues maybe a year and a half ago, something like that. So the field of rhizosphere biology kind of disappeared, for a couple of decades. And I think that the agronomic community as well as the wild plant community are very interested now in the plant microbiome. So I think we're going to start seeing that coming back. So perhaps then you could fold in the toxicology. But I think this community is really wrestling with a lot of difficult issues because, for example, the takeaway from yesterday, for me, one of the takeaways was that even your population, the humans, are changing over time, right? So it's shifting baseline that way. Plus the environment is changing as we have new pollutants or new toxins or global climate change. You can look at any scale you want to. That's changing also with time. So it's a really, really difficult field area that you're wrestling with. There's no doubt about that. And when I've talked with medical doctors, they also are starting to recognize that the diseases that are named IBD in the 40s was a very different disease than IBD is now, inflammatory bowel disease. So even the medical field is wrestling with the notion that what we call cancer, or what we call IBD or whatever, is also changing. So I think that's a common thread throughout all of these areas when we're dealing with human health, is it's not a fixed property either of the subject or of any of the things you're trying to measure. So that's really tough. However, I do not want to come off as advocating for collecting every single possible property. I mean, if you were to take on a major initiative to collect RNA, I think you would fail and it'd be expensive. It's a bad combo. Because messenger RNA, I told you DNA is nature's most stable molecule, biological molecule. Messenger RNA, by definition, is nature's least stable molecule. It has to only live for microseconds or seconds. So all these cool kits and so on are not going to get you good data. So I think I do want to offer a cautionary note. I'm not advocating that every molecule be collected, but again, like the thing I asked before, make sure you know what your question is, and then try to proceed to collecting the molecules that you need in order to address your question. But boy, RNA is tricky, and I think that would be the last thing on your. It'd be RNA the least, protein the next least, and then maybe we'd go after DNA and metabolites. Might be the two kinds of large molecules that would give you the most bang for your buck, and hopefully the most informative. So I think we want to temper that. There are, by the way, a number of communities that are trying to develop standards and trying to figure out what's a SOP, a standard operating procedure for doing these analyses. A paper just came out a couple months ago, maybe at most, in I think it was in Genome Biology, and I think the lead author is, I'm going to look it up, Sinha. It's a working group that formed about 2 years ago called the Microbiome Quality Control Working Group. And what they did is they did basically a round robin of about a dozen different labs, and they asked the question for DNA analysis, what part of the entire pipeline from sample DNA extraction to analysis is the most fraught with error. It was an excellent analysis. And they published the initial results as only a pilot study in Genome Biology a couple months ago, and I'll send the reference so it goes into your report. So there are groups that are super interested in developing standard operating procedures for all the different types of molecular analysis that we want to do with the microbiome. And again, I want to reference the NIST standards workshop. I think it's kind of wide open as to what they're going to address. So if anybody's interested in attending the NIST workshop in April 4th is a Monday, I think, let me know and I can send you the link to the workshop. But that's sort of how I want to address the other comments.
So Sam Forey from NIST, that workshop is, April 4th and 5th, right here in town in Gaithersburg. It's standards for microbiome measurements. It is going to be a working meeting. We would really love a lot of help in determining what the most important needs are for moving this community forward and supporting some of the really cool measurements that we've heard about and some of the connections that are being drawn over the last couple of days. So if you have questions, I'll be here in the audience. Please feel free to ping me, or Kelly Rogers is here. And of course, everything is available online. It is an open meeting, and we'd love for everyone to participate.
Could you give that information to Keegan and Marilee so that it goes into the report?
Absolutely.
Thanks.
Thanks for plugging it, Lita.
Of course, of course.
Thanks. So one of the things that
And maybe I could just follow up on what you were saying about USDA. My name is Isabel Walls, and I work for USDA. We are looking at plant microbiomes. We're looking at the viruses as well. And we're looking at what the normal microflora might be and how that might protect the plant against plant pathogens and human pathogens. So you may know that leafy greens are now the biggest single source of foodborne microbial disease. You may not know that. But, Estech E. coli and salmonella on leafy greens are now a very significant problem. And of course, you don't cook lettuce, so it's not like chicken that you can cook fully. So, we are starting to look at this and also the genetic factors that might affect this as well. So, but it's a very small amount of money. We really wish we could be doing more.
So, but Isabel, it's my understanding it isn't just about foodborne microbes on plant. You're also looking at the endogenous microbiome of the plant, right?
There's a bigger piece to it that one of my colleagues is working on that I can get you more information if needed. I'm the food safety person. I just want to give a plug to. When you're looking at environmental exposures, I'd be very grateful if you include microbial foodborne pathogens.
And then with regard to the phytobiome, if you have anything more, could you provide that to Keegan Merrily? Good.
I wonder if I could just make a comment, which, because I like spoiling people's days, you know. Everything that I've heard today in this whole workshop has been really interesting. It's been about the microbiome. But the population, you know, the ecology of the gut is not just bacteria. So, you know, we just got there viruses, you know. It's not just viruses, it's parasites. And anyone who's had a mouse colony has got parasites. And it's also about yeasts. So one of the unrepresented populations that you really need to get hold of is a load of virologists who can tell you about the viriome, because it's really quite important too.
I absolutely agree. I'm going to admit that the HMP really zeroed in on the bacterial component of it, because it was the most, thought to be the most abundant at the time. Of course, papers have since come out to show that viral particles are 10 times more than the bacterial membership. I think we're starting to see some emergent viromics work. The report I referenced actually calls out that it's a severely understudied area.
Yeah, I was actually going to make a similar comment, because we heard a little bit about that yesterday, and a discussion about the fact that certainly even eukaryotic parasitic organisms were something that helped us evolve the niches that we're talking about here. But I do think that at this point, as we begin to look at function, we need to keep in mind the fact that these other organisms are there as well, and may have a role to play in that function. Yvonne.
Yeah, I think it's these are not to be overlooked. But again, I think I'd like to pare down the expectations of, wow, there's other worlds out there and there are galaxies to be discovered. I think this starts sounding like astrophysics to me, which is great. But at the same time, I think equally important are the mundane process of decision making. And I think a great example of how the field of toxicology and environmental health has really embraced the systems biology in the last decade or two, and the reverse is not as exciting, because in reality that all of that knowledge hasn't really, percolated or taken strong hold in how we make decisions yet. I mean, there are some good examples of that, but we haven't revolutionized decision making along the lines of the biology and the science. And not that we want to regulate or make decisions on the coolest science that just came of age in the last 5 years, but at the same time, I personally do not believe that that gap is the easy one to close, and it would naturally follow that the leading edge is the science and the trailing edge is the decision making. I think that gap is widening rather than closing, and in that sense, I think we would be remiss to point out, while there are exciting horizons in biology and microbiome, virome, and yeastome, and whatever, I think we really need to make sure that the scientists who design these studies, the epidemiologists or toxicologists who would conduct these studies, not only think about what they would collect, but also at which point of entry along the decision-making continuum that information would actually make a difference or not. And I'm personally not, as optimistic that knowing more would actually lead to necessarily quickly better decisions. So I think we ought to talk about, which part of the decision-making process, microbiome and other related science, is going to make an impact. Is it on the better characterizing hazard, better understanding dose response and metabolism, better understand the vulnerabilities of individuals who may have, different microbiomes or whatnot? And I think the quicker we understand where that point of entry into fairly linear and established process of decision making is, the better we can communicate this information to decision makers.
Yvonne, are you often the designated driver? Because you're always very good at sobering the conversation. I know it's great. It's great. It's not a criticism, but I just thought he must be the designated driver in his social group. I'll bet. So so there you go. So I'm going to harken back to a personal experience. When I was in the field of marine microbial ecology, it was, date myself, far before there were any molecular tools. But that was actually a good time, because we knew microbes were important in oceanic processes, but we had no really good way of directly measuring them. So modeling to develop a hypothesis about what the role of microbes could be was critical to advancement of this field. And a lot of progress was made in the field. And then came the molecular tools to figure out what community composition and so on was. I mean, it added quite a bit of knowledge, the use of molecular tools, but I don't think it fundamentally changed sort of how we thought about oceanic carbon cycling and nitrogen cycling. So to my mind, modeling, hypothesis building, testing is probably what this field needs when it comes to how you want to think about the microbiome. And the second comment I'm going to make probably also dates me. You know, there's a whole generation of students now who don't actually do an experiment because all they have to do is mine data, right? Which, I mean, it's a whole new way of doing science. But the thing is that there is this sense that there's so much data out there that they can actually develop hypotheses by just looking into the data. Now, maybe there's a way, though, that more modern approach plus model building can be partnered to figure out for this field, what are the key things you need to ask in order to understand the role of the microbiome in environmental toxin exposure?
Let's take some questions. Tina, we'll start with you and then Joel.
Okay. So Tina Bahaduri from USCPA. I want to push back a little bit on what Yvonne said earlier, in that I think decision-making is for the sake of making better public health protective decisions, at least focusing on health. And I agree with you that there's also a commensurate and probably actually more advanced effort in terms of the eco space. When we submitted reports to that FTAC, most of the EPA's work in the domain of the microbiome was actually through the work that's being done by the ecologists, because the ecologists don't actually separate all these stressors as if they're independent. They look at interactions. They've always looked at interactions, and they've always dealt with complexity, and they've always looked at a systems approach as opposed to humans that seem to decide somehow that these stressors are all independent of each other. So, Lita, I think you're right that there's a lot going on in that domain as well. But focusing on the human health, the funky title for this workshop was one of the contributions that I made, because I really came to this workshop needing to know, does it really matter in terms of public health protection? Are we, as you're suggesting, Yvonne, our current approaches intentionally or unintentionally taking into the account the role of the microbiome, and therefore our decisions are adequately health protective, which seems to be what you're saying, that knowing more would not have changed the outcome of our decisions, or knowing more would have changed the outcome of our decisions, because there is clearly some disparity in risk assessment derived levels and what the public health community is telling us about whether decisions are adequately public health protective. And what John Vandenberg said yesterday is this begins to indicate that maybe the microbiome can explain some of that variability and some of those things that we're not capturing in a classical risk assessment. And I don't think that we can afford to wait a year and begin to collect the data. We heard yesterday from Germaine that what she heard is that this is infinitely doable right now. So I think it would be wrong to wait, but probably more think along the lines of what Margaret was saying, or what NIST is doing, is start coalescing around methods, approaches, data types we need to collect. How do we start not splintering into a million different directions, but start to coalesce up front so that we can do something meaningful? And I completely agree that this is going to be expensive, which is why we have to be very thoughtful. But I really, my walking away message from this workshop is it seems to matter. We don't know how much it matters. We don't know if it would have changed our past decisions, but it seems to me that it might change our future decisions as we're burdening the system more and more. As Margaret says, it's the range of stressors are increasing. As we heard from USDA, that there are stressors in the environmental system that are going to become human health problems, whether we're looking at that or not. So it just tells me that there's a compounding effect, and we need to have data to know what the heck to do with it. So I wouldn't postpone, I wouldn't pause. I would just have to think about how to do it, really.
Yeah, so Tina, I think we vehemently agree, and I didn't wish to paint as gloomy picture of the state of affairs as was painted in last night's presidential debate, but. I think I speak from personal frustrations, which then started shaping some of the things we're doing and our collaborators are doing. I think a lot of scientists, my comments are encouraging to scientists to learn about the process of decision making, because a lot of us who really haven't been taught this in graduate school don't realize how the beautiful ship, shiny new omics ship just crashes into the iceberg of decision making, and we want to prevent that. And my suggestions were not to wait and delay sample collection and study design. My comments were more on encouraging the the Human Microbiome Project and other large scale projects that were not designed for environmental health but were designed for human health and microbiome questions, to really quickly answer questions. What should we measure? It's not to delay sample collection, but not to just plunge, jump off the cliff into let's measure everything. I think that would be prohibitive, and also it will not allow us to communicate that information better, because as as John knows, decision makers not necessarily looking for too much information. They are looking for right amount of information, and that allows us then to communicate it much better.
Thanks. Good comments. Lita.
I wanted to respond to the comment about
Closer to the mic, please.
Oh, sorry about that. I wanted to respond to the comment about cost, and that I don't want this community to feel like you're alone in this. Part of the motivation, obviously, for the FTAC-MM workshop or the committee was to identify common gaps, needs, and challenges, and then figure out what we can do to try to meet them. And that was across the federal government. I don't know where this is going. We were formed for the express purpose of providing information and data for the president's fiscal year 17 budget request for a microbiome initiative. I have no idea if that's going to come off or not. But it involved the private foundation communities, involved funding agencies, it involved the external community, involved many, many different stakeholders. So at least they started the momentum going on this. And so I think the idea is you don't have to reinvent the wheel in this community, and neither does NIH, but there's going to be a need for common resources, databases, computation. Exactly. Standards, technology, computational tools. There's a lot of common needs for this field. And also, we're producing huge amounts of data. There's got to be a way to make sense of it, to be able to integrate it and look across properties. So those are the kind of themes that came out of the FTAC. And I'm really hoping, Yvonne, and everybody here, that something does come of this effort so that this community, as well as other communities, can benefit from it.
Let me just respond to that comment a little bit, because I think it's important for people to recognize that we had a very interesting experience with the nanotechnology initiative. And all of the agencies were doing nanotechnology work. And the approach basically from the OSTP was to get those agencies together, to catalog what it was that they were doing, and to begin to look at how the field could really move forward. That was very difficult, because each of the agencies were making decisions on their own budgets and figuring out what they were going to do. And it was difficult to listen to another agency tell you what you should do with your money. And so we did 2 reports out of the National Academy on the directions for nanotechnology research that really urged the agencies to come together and really think about this. And as a community, it's up to us to try to continue that kind of a theme in this emerging area as well. And so I'm encouraged by the work that's already gone on, but I think that it's going to be very important for us as a community to get back to our agency colleagues and talk about the kinds of things that we talk about at these meetings. Joel.
Thank you. So I wanted to address a couple of points that came up. First, I think that what we're seeing with this emerging area is that some of the naive concepts that we applied for doing risk assessment, don't really work. So that, as Yvonne mentioned, we have this notion that, well, there's dose response and then there's susceptibility, for example. Well, if all the dose response curves are linear no threshold, then that's true, right? Those are separable questions. If they're not, those are not separable, right? And they're probably not, right? So I think we need to sort of move to somewhat more sophisticated ways of thinking about it, and this is an area which may help us with that. And so I think that's very good. The second point is that from the point of view of public health about environmental issues, things are different than from the point of view of personalized medicine. And that actually means that we can extract useful information, I think, sooner. So let me give you an example. Suppose I think that one of the reasons for heterogeneity in response has to do with the microbiome, right? So that the microbiome modifies my response to an environmental toxin, say by metabolizing it in the gut or whatever, changing functional forms. And then I don't have to know what species are doing it. I don't have to know what metabolic profiles of the gut microbiome are doing that in order to help me do my risk assessment. What I need to know is what's the distribution of how much modification I'm getting, right? What's the 90th percentile and the 10th percentile? Because now I can do a risk assessment and I can say in response to ozone, for example, and, here's what the average response is. My studies have already told me that, right? But here's what I think in the humans the distribution looks like, and here's what the 90th percentile of response is. And so now I can improve my risk assessment by saying, okay, this is how people respond to ozone or lead or some pesticide. And if I'm worried about the people who are being impacted the most, I have some estimate of what that highest response is. But the microbiome is not just a modifier of response. It's also potentially a mediator of response, right? Because environmental exposures may change the ecology of the microbiome, right? By killing some and favoring others, etc. And so again, that's an area that will take us probably decades to understand, but it will take us a lot less time to, again, try to understand how much, right? So what is the 90th percentile of the change in the microbiome and the 10th percentile and things like that in terms of its metabolic functioning and therefore its potential impact on human health? And so from the point of view of trying to make public policy by having good risk assessments that take into account the diversity of responses that we get, I think we're in a position to improve that substantially by looking at the microbiome, by not so much focusing on understanding all the details, although we want to do that eventually, but we can get useful information out much earlier if we think about it that way. Thank you.
Thanks. That's a good perspective, and I think it really gets at the kind of thing that Yvonne was talking about. And we talked a little bit about that yesterday. We're not at that point yet in terms of being able to put those bounds around this. But from a functional standpoint, it is something that we can begin to address relatively quickly. Anne?
Yes, thanks. again, to Yvonne's point, which echoes one that Sean Eddy made about 6-7 years ago, along the lines of when will we stop sequencing genomes? When do we know enough? I haven't heard any hard answers to that at any point from any sector, but I think that one of the things I want to offer a simple heuristic that might translate some of the stuff that you toxicology, epidemiology talk about, as the previous speaker did, that are kind of like, okay, Greek to me as an academic. But there is a lingo in academia that has also been brought up that might be a simple heuristic for grant proposals, for example, or for solicitations. And that includes having the testing. We you know, Lita and I and others with gray hair in the room grew up in the era of you must have a hypothesis. There has to be a hypothesis in your proposal. There has to be a testing of a hypothesis in your paper, okay? And then all of a sudden there was this explosion of descriptive, what we used to call natural history in a derogatory way. But natural history is what we're doing. And so it's okay, you know, to do natural history right now. In fact, we're, you know, swimming in it. So, but we don't know what to do with it. So I think that a return to the requirement in this context of a hypothesis, each proposal must have a hypothesis. If it's for nothing more than why or how or what you're going to find when you do this, it's got to have a hypothesis in it. And an alternate hypothesis, and I won't go into what that means. It doesn't mean the negation of your hypothesis. Okay, and the other thing is this is the importance of modeling. This is where modeling coming into this field, NIH has got to do it. DOE is doing it wonderfully, and it has been brought up earlier. DOE is supporting all kinds of modeling right now in biological systems. And so I think modeling is the other thing, because that's a way to develop hypotheses that are testable. That's the whole point. At least in, I mean, from a regulatory point of view, it's a different point. It tells you risk analysis, but from an intellectual context in developing experimental science, that's what you use modeling for. And I think these two things need to be key elements in every proposal, every FOA that goes out. And I think that will naturally tend to help us focus on the doable, which is a very worthy goal that will be the biggest bang for the buck. So.
And I think, you know, I think that's exactly what we were hearing from Joel in terms of the kinds of things that might be appropriate in terms of putting these kinds of things in perspective. And Michael Broder made a similar comment earlier in speaking with Lita about the importance of thinking this through from the standpoint of a hypothesis that could be addressed, and then looking at modeling as a way to sort of put perspective around that hypothesis. I think that's a great way for us to think about this, sort of the state of the science in this field right now. Yeah.
It's more intelligible to academics than the risk analysis stuff for non-professional listening.
Sure, yeah. Applicable to both, though.
Risha Putzrath, Navy and Marine Corps Public Health Center. Insert government employee disclaimer here. It's especially important today because in the spirit of Jonathan Swift, I'd like to make a modest proposal. We've heard a lot yesterday and some today about the need for interaction among different disciplines. And what I'd like to propose is something that I'll call, for want of a better term, a Craigslist for research. And the idea would be that as we're talking about modifying how proposals are written for federal funding, that one part might be that each proposal for research have, in addition, I don't know, offhand, maybe 3 areas where they think that additional information could be gained from their experiment, but they don't have the expertise. And playing off of one issue that was just raised, we're going to look at the microbial community in the microbiome, but if someone wants to look at the yeast, we don't know how to do it, but we can provide you with a sample. Alternatively, also with the modeling, we know how to do basic statistical analyses to see whether the results are different. But if you'd like to use our data to do some more sophisticated modeling, we'll work with you in designing our experiments as we're doing them, if you need some different information that's easy for us to gather at the same time, we'll gather it. And I think, the internet has not provided, at least in some of my areas that I would be interested in, this great community to get information around quickly. But this is one place where I think it might be useful and be available to have different labs that don't have collaborators in the other fields. Some of us do, some of us don't, be able to pick up a collaboration and have some place where all these would be posted and someone could say, oh, that's great. Or I could have a graduate student do that for a thesis and tap into that.
Actually, I made a comment about the newest generation of computational scientists, but actually, I mean, their whole desire is to take new data sets and see if they can actually, they're not just exploring, they're trying to test a hypothesis with existing data. So my question to this community is, all the different data that you produce, I mean, are they rapidly and broadly advertised and disseminated so that people who are computationally inclined can actually get at the data? We're having that problem at NIH, where we have some, very well-known repositories, but the structure of the repositories make it sometimes quite cumbersome to try to actually extract data from them. NIH is now trying to move to this NIH Commons model using the cloud, hoping that will actually make the data more available to the community. So my question to this community is, what are you doing to get the data out there so that people can chew on it? Because I think it kind of ties nicely to your Craigslist question.
Well, the thing that I'd like in addition to that, because sometimes you can get the data, sometimes you can't, that's always a individual researcher prerogative to put the data out or not sometimes. But all of us who've done experiments know post hoc, if somebody had raised the issue of, again, taking the same example, looking at yeast in addition to the prokaryotes, or looking at phage in addition to the prokaryotes, that it might not take much more work while you're doing the experiment to get those data that somebody else could use, or to send the samples to somebody who could get those data as part of the experiment. And so the idea that I have is not just to make the post hoc data available, but to let people who are interested in the field know that you're going to start an experiment that's going to look at X. Is there another data point that we can gather for you if it's an epidemiology study that would be dovetailing with your research? Or if it's a laboratory study, is there a sample we could send you so you could gather data that we're not going to be looking at? And I'd like to see it not just the data after it's been generated be available. That's one issue. But can we set up collaborations as the study's starting? Because, as I say, some of us with gray hair know a lot of people in the field. A lot of the beginning scientists don't. And this would be a way for them to make the connections and get even more information out of one study.
So, Risa, I think the proposal is an excellent one. First of all, at this meeting, I've heard at least 4 different investigator pairs talking about getting together to do some research, feeding off their presentations here. And I think, and you and I go back a long time, and so we've watched this, and John, you also, I don't think there has ever been a better time for collaboration among federal research. That doesn't mean there isn't a lot of room for additional improvement and collaboration that could go on, but this is doable. And the kind of thing that you're suggesting is something that there may be ways to stimulate that, and that's what we ought to be looking for. Yvonne.
I agree, and I think that the onus is as much on the funding agencies as it is on the investigators, because the program officers and staff at extramural research programs at the institutes and centers are probably the best knowledgeable about all the different eligible bachelors, and so the matchmaking function that I know many institutes are taking to heart is a very important one. So I think it's I'm not necessarily sure whether I need another internet resource to I think I need less internet than more internet. But I think importantly, building these partnerships and building these consortia and encouraging funding opportunities that really facilitate these interactions. I know NIEHS is moving in that direction where they're trying to not instigate things, but they're trying to facilitate. And I think those funding opportunities really can do much more to move science faster and fund the same number of investigators rather than just necessarily one-off R01. So I think there has to be balance there, but the institutes and centers have a large role to play as well.
I think there has been a real push toward leveraging of funding, and romance aside, I think the idea of leveraged funding is something that all of the program officers are beginning to think about now and look at as an opportunity.
As long as someone is watching the leverage buyout level and and the risk in that market. So.
John Balbus, I'm Senior Advisor for Public Health at the NIEHS. I have two comments which at this point in the line are building on some previous comments. First, building on Joel's comment a little bit. I know that the focus of this workshop was on the functional aspects of the microbiome, primarily as an effect modulator of environmental exposures, but I do want to keep a broader perspective on the idea of microbiome as mediator of some very important public health impacts on cardiovascular disease, on inflammatory bowel disease, on asthma. I think as we get a better understanding of how the microbiome is a primary driver of some of our biggest public health problems, we need to go upstream and look at what are the environmental factors that are driving changes, dynamic changes, or the substance of the microbiome. In much the same way, we're just starting to scratch the surface at the effect of the built environment on physical activity, which is shown to be a primary driver of colorectal cancer. All of these things that if we want the impact of public health, we also have to look more upstream at what are those drivers. And I think we need to be taking a One Health approach to that because we may find that there are hotspots of diversity, hotspots of change in the microbiome if we look in a focused way at that. And it broadens the decision space outside of just environmental chemical risk assessment. The other thing, I want to come back to this point about modeling and the idea that it's not in the culture of NIH to model. It's true to some extent, although there's certainly a good deal of transmission dynamic modeling going on in infectious diseases at NIAID. It's opening up a little bit. But at the same time, I think we need to change that because, I work in a community that does a lot of modeling climate change and health, and the Department of Energy is driving a lot of this, and they are desperate for people with really strong, rigorous health credentials to be able to assist them at that tail end as they're modeling the upstream drivers. And a lot of the people that I work with, put their grants in, their proposals into NSF, and they get told, well, no, that's health. We hear that so much. And so we need to have a home at NIH for this. I think that as we live in a very dynamically changing environment, observations are not the only way we're going to be able to inform decisions. We have to be able to model for predictive changes in a dynamic environment. So, two editorial comments.
John, I think the discussions that we had yesterday, particularly about diabetes, TMAO was mentioned, a number of those issues around how the microbiome actually does have implications for public health and disease from that standpoint, with or without an exogenous chemical agent, in the case of arsenic perhaps, or ozone is another example. But I think that it's an important aspect for us to think about, because we've got the public health dynamic that comes simply with the fact that we have a microbiome, as well as the discussion about how that might mediate or modulate the exposures that we're seeing. Margaret?
Yes, this is a point I wanted to bring up too about the microbiome as a mediator. And what I presented during my talk illustrating arsenic in the model where I showed, these are the factors that are driving the developing microbiome and then in turn disease risk. And when we're talking about modeling, it's not maybe atmospheric modeling or oceanic modeling, but our epidemiologic models need to consider, as Joel articulated, is this an effect modifier of susceptibility to an environmental toxicant, or is this a mediator? And this will affect our statistical modeling. And as we think about where do we go now in the future as we are accumulating data, I think an emerging area needs to be how do we model the microbiome as a mediator and potentially invest in developing the bioinformatical and statistical tools that we need in our studies to move the field forward. So I just wanted to throw that out there too, as an area that needs to come with us as we're designing our epidemiologic studies.
Yeah, thanks, Margaret. And that gets at our second question here about discussions around advances for epidemiologic and occupational studies. And I think that's something that we want to continue to look at as well. Please.
Hi, Isabel Walls from USDA. So just referring to the question earlier on what is the role of the microbiome on dose response? There's some interesting data that shows if we perturb the microbiome through use of antibiotics, and then you challenge somebody with salmonella, the dose response is much lower than if they have a normal healthy microbiome. And so clearly just looking at the microbiome itself and what impact, I'm sure that's true for environmental chemicals as well, that there is a direct impact of the microbiome. That's a very researchable question, and we should be able to do that quite easily. But I think we cannot forget the questions that John Balbus just raised. And there's some very interesting research coming out of the nutrition community that's looking at the impact of diet on the microbiome and the changes, the functional properties of the microbiome. So when you eat certain foods, the bacteria in your gut will change and they will produce different metabolites. So these metabolites could be carcinogens and they're having a functional effect. It could be on colon cancer. But we've also heard about the microbiome producing chemicals that affect the heart. And they're having a functional effect on the heart, or they're having a functional effect on other target organs. And to me, this is the most exciting part of this research. And we need to move quickly, because I think if we look at the role of prebiotics and probiotics on the microbiome, the functional properties, the metabolites being produced, and what is happening to them, what are they doing to us, I think is going to resolve a lot of the questions we have on chronic disease. And it is so exciting. And I wish we had more money.
Please.
So I'm Carolyn Mattingly from NC State and a member of the committee. I just wanted to make a comment about outreach on different levels. So I'm not a microbiome researcher, but attending this meeting, I now have this like, oh crap response to this. Like, what do I do next and how do I model this? And so we're all kind of preaching to the choir here, but there are a whole bunch of people out there with funding that are not thinking about the microbiome, and through no fault of their own, but just that it's difficult to, I think, acquire an appreciation for how important this may be. So I think we have to think about how to access that community and come up with, you know, in the absence of the people who are actually doing this, knowing what to advise, some sort of guidelines, because there's research where I think there could be some missed opportunities unless we interact with them. And then the other scale of the outreach, and this has been touched on a little bit, is NIH has lots of really big research programs that do spectacularly well working by themselves in silos, things like the BD2K program, the Exposome program, the new initiative with CHEER, and I'm peripherally involved in some of these, and I don't hear much discussion about microbiome in any of these programs. And particularly with CHEER, it just seems like this would be a huge missed opportunity to not get involved in that. And for the awardees who are involved in that and are charged with processing samples, they're very focused on metabolomics and metabolomics, and there's no reason why that couldn't or shouldn't be expanded to include some microbiome processing and coordinating with some of you folks who actually have expertise in processing some of these samples.
So I'm at NIH and I don't know what the CHEER program is. Could you tell me? No, that's actually it supports your point, but could you tell me a little bit about the CHEER, just even what the acronym stands for?
Take the mic. Yeah, I was going to say, please come back. You'll get close, though.
So it's the Children's Health Program that was closed, and they took those funds and...
I thought that was called ECHO.
Yeah, no, that's actually, it's sort of related but different. And they're not really talking to each other either. There you go. Children's Health Environmental Exposure Analysis Resource. So it was this sort of three-tiered program where there's a coordinating center, which was awarded to Westat, and then there's a data science center, and that was awarded to Mount Sinai to sort of coordinate all of the data that would be generated. And then there were several universities with expertise in things like metabolomics and metabolomics, and the idea was that people who are involved in children's health initiatives, epi studies, would have these resources. They would sort of have these mini applications where they had cohorts, they would have samples, send them to these centers that would process the data. Those data would then go into these data repositories. So the idea is very interesting in that it's trying to coordinate a lot of cohort-based initiatives that are looking at children's health and environment, and focusing on phenotypes that are manifested through metabolomics. And in a lot of cases, the exposures tend to be metals. So there's also capability with metabolomics. So, sure.
Yeah, thanks, Carolyn. I think, the good news is that we've made a lot of progress since our first meeting 5 years ago, and we're seeing even in terms of attendance on the webcast and here, the kind of response to an opportunity to hear about these kinds of issues, particularly with regard to the microbiome and environmental health. There really have not been a lot of meetings around that, and this is a nice opportunity to bring people together. But I think listening to the progress that the HMP has made and the kind of things that we're doing, we're getting there. And talking to a couple of the centers that are part of the CHEERS program, they're very aware of the microbiome activity and the need to try to bring those data into the discussions of the metabolomics that they're doing. So my sense here is that it's encouraging, but we've got a ways to go. Yeah.
Linda Winterberg, I'm from NASA. I'm also a liaison to the committee. Fabulous set of talks. Was blown away by parts of it that I could take to my daily life and reflect on the fact that I can thank my mom how she raised me, because it probably made good things for me over time. But one of the thoughts I had was, when I listened to this, were two. One, which was, and very much my colleague who just spoke before this, there are a series of things happening across the federal government. There are massive databases. On this committee, one of the liaisons represents the folks from DoD that have huge amounts of information that they take from people who are in the services. A lot of information, they take a lot. This is the sound of someone who is not a person doing microbiome research. Is there a sense of the kinds of available, even federal or even international databases, that could provide good basic information to this, so you wouldn't have to design every study and do it all over again? And that's just a call to everyone to think about that. Is that there? I mean, when I was at DoD before, I got very aware of the whole service person thing that they were doing, where they were tracking all kinds of information. It may or may not be useful to this group, but the issue is it's not something that often folks in the practical research world see. So there are lots of, I'm just saying the fact is there may be resources a lot that we have never actually put in one place. For example, if we're talking about programs like CHEER and half people are going, I don't know what CHEER is a very reasonable thing. There's just a lot of work going on. There's data repositories in that. I didn't know about those. Those are the kinds of things that as a pragmatic person to promote environmental health issues, if we have data that we could use,
we've got to start really finding ways for the microbiome or the exposome or any of the things we've worked on to try to link that up. And so that's a challenge to the committee or the community to think about how we could do that in some sort of consolidated way. And the other one is just a personal statement. We're talking a lot about lots of information. We have lots of data. That's my only sobering statement. We have a lot of data. We don't necessarily have a lot of information that's integrated away, that especially information that can be used in decision making. And respectfully, that there are reasons why that's happening, but the wealth of data all over the place is actually a detriment for us sometimes to actually be able to communicate this effectively. So that's another challenge to the community to think about that. But otherwise, fabulous presentations.
Thanks, Linda. Neha?
Neha from UCSD. Just referring to the comment back about public availability of data sets, I wanted to bring your attention to two of the servers that we have. One for the metabolome, which is GNPS, which is not just a repository of data sets, but also does automated analysis of the available data sets. Also compares different data sets from different habitats that are available on the server, which allows you to find commonalities and differences between habitats. And for the microbiome, there is Cheetah. These data sets also provide some of the statistical tools that are important for analysis, and we are building upon more of these tools. So we really urge the community to deposit more and more data sets, because this allows us to build these statistical tools that are important for analysis. Thank you.
Thanks, Neha. And I think, getting back to what Linda has just said, without the tools that you're describing, that's just data. And so the opportunity really to begin to look at how we develop not only the data sets or repositories for the information, but the tools to do the kind of analysis or data mining that we've heard about is going to be particularly important. So, we're sort of working our way through these topics that for discussion, and we've said a lot about design characteristics and reporting so that the implications are clear. I think that was certainly Yvonne's comment about trying to make sure that we're efficient in the way that we look at the way the field advances, particularly with regard to application or use of the information. There's another aspect of this that has to do with sort of the institutional imperatives that might help this field move forward. And we've heard a fair amount about that today, but there may be some other things that people want to say about that. We've heard about the idea of encouraging the collaboration, of building the various repositories and tools that would be available, and the help to do the matchmaking and those kinds of things. Are there other areas from an institutional standpoint that either our committee members up here who are part of the panel or people out here would like to comment on? Ann?
Take the panel first, that's fine. But that's exactly what I was going to speak to.
Go ahead.
Monetizing the microbiome. Is anyone here from pharma? Anybody from pharma? Anybody from big ag? I mean industry, okay?
Close enough.
Yeah. What I think is important is that this area has grown so rapidly, and it's certainly getting the attention. Pharma, in terms of pharmaceutical, intellectually it's on its uppers, basically. It's gotten to the point where they're actually talking about and sharing their clinical trial data from company to company, appearing naked before each other. And so that's where the money is, more than it probably should be, but that is where the money is. And when they get into things, they dominate the market. Computer science graduates are not going to, not finding jobs in academia, they're going to industry. And I think that the same thing, crystallographers can go to work in industry, but many of the big centers of crystallography aren't hiring people anymore. That are funded by government grants and so forth. So the question really is, to what degree is pharma, do we know, engaging in the microbiome, and to what degree can we enable them and encourage them to engage in and appreciate? Because many of them, and also the federal government has spent hundreds of millions of dollars on clinical trials and a more abstruse concept, GWAS, genome-wide association studies, that have brought home, you know, huge wide swaths of potential genes. Maybe 1-2% of this one and that one, and you know, and so they're failing, and they are getting, you know, I mean, it seemed like a good idea at the time, but I think the two major reasons that they're failing to have concise answers, and that is clinical trials do not frame their subject populations adequately. They need to know, I've said for decades, they need to know something about tox exposure in order to pare that down so that they can figure out what subpopulation. They want to make a drug that'll work for everybody, of course, but they'd like to make a drug that'll work for somebody, and I think that's increasingly a problem. And yes, it's important to know about the role of the microbiome in creating and ameliorating side effects, and some of that obviously is industry would like to collect and know that data, but just to be able to know something about the microbiome and also about toxic exposure, I think would enable clinical trials to be much more effective and return the bang for the buck that they're putting in. The same goes for GWAS studies. And so the real question is, how can we get more people from big pharma to meetings like this? And maybe they left already, I mean, because we're not the full crew. But I think it's really important for us to do that. And one of the vehicles for kind of bringing industry and academia together are the Gordon Research Conferences. And I think I saw something about it, that there's going to be a microbiome Gordon conference. Do you know, Lita? Okay, so that was just the other piece. But this is just, if we can get industry into this, there's a risk if we get industry in, because they like to keep their data secret. But I think they might realize, having already begun to share their clinical trial data, that they need to share this kind of data as well. That's all.
So let's hear from Ian and Lita on this topic.
Okay, well, pharma is very well aware of the microbiome. There are a couple of companies that are investing in it in quite a big way as a druggable target. Toxicologists in pharma have been aware of the potential problems of variable microbiomes for a while. I'm not sure there's vast amounts of money swirling around in pharma to spend. I have also noticed you haven't got anybody from industry here that much, and I think you need to reach out to them because they will be interested and they'll work as partners. Whether they will fund you is something different, but they will probably want to work as partners. And they also have, I think, a whole range of very useful tools that they can share, because they have lots of very bad antibiotics that are not broad spectrum, they're rather narrow spectrum, that can provide us with really useful tools. Rather like Matthew's, let's knock out deglucuronidation. Well, let's knock out some of these species and see which are the keystone species. I'm sure you can get some very good collaborations going.
I've always been sure that we could too. I haven't seen it yet.
But you have to ask them to come. Now, I can help you with some names.
So Ian, I'll probably address it to you as well as the whole group, but lately, maybe in the last few years, I've heard discussions about the interest in pre-competitive consortia. So the idea is that you mentioned money. I mean, every company, even if they're a multinational company, can't afford to run all the experiments themselves. So they want to create sort of a set of experiments that everybody needs to do in order to get the foundational data, and then at some point in time, based on memorandums of understanding or whatever, they step away and then do their own proprietary research. But that pre-competitive consortia model is meant to open up the community that could do these experiments with academia, with industry as partnerships. And I've heard a lot of that discussion happening, particularly around the microbiome area. Johnson & Johnson, AstraZeneca, GlaxoSmithKline, all those really, really large pharmaceuticals are super interested, of course, in drugging the microbiome. And there's a separate industry, nutrition, food industry. They are on top of the microbiome. They're trying to figure out some way to design foods to benefit our microbiome. So I want to hear what you think about this pre-competitive consortia model.
I'm sure it can be done. I think it needs leadership. I think the leadership has to come from you guys as academics in the first instance, say this is an opportunity that we'd like everybody to get dragged into. Well, yeah, I could take some of his money. You know, there is an interest and an eagerness, but pharma is also Sorry, these are opinions of mine, by the way, and they're my opinions. They're not anybody else's. Pharma's in a very bad state at the moment because even if we invent the drugs, no one will buy them. So consortia are great. I think there would also be a certain amount of fear in the regulatory area of pharma that digging into the microbiome will simply mean that they have to do a whole load more of expensive testing. So I think there's an opportunity here, and it has to be carefully handled. Definitely an opportunity.
Matt?
Yeah, so as a canary in a coal mine, I've been in front of a lot of big pharma to talk about this, and I completely agree that they're very, very much interested. But, they're very practical. And so, talking about big ideas and big consortia, there is no way that a good industry representative would get involved in this until there is a very good reason and a concept proved. So, when I was a lad, kinases were relatively new, and people could barely believe that a chunk of inorganic matter could be added to proteins and that could have a difference in terms of human health. And now, of course, every pharma has just a vast amount of effort put into this, and now epigenetics and so forth. And all of that is very Darwinian. It does not come to fruition until it is a proved case. And once there is a proved case, there will be that pent-up desire and that sort of figuring out and kind of throwing elbows, how are we going to do this, it will burst forth. And so I would just caution that, and we should definitely reach out to them. So I don't know about a Gordon conference, but there's definitely a Keystone meeting on the microbiota, and that's where I've met a lot of pharma people. They are certainly interested, and they're very bright and ready and sort of very much understand this, but they can't walk into their upper management and say, we need $350 million, and we promise it'll work, and so you're going to say yes, right? No, they're not.
Put a plug in for a meeting. I don't attend them, but it's that environment. They're called Microbiota R&D and Business Forum meetings, and they're being held all over the world. I think this year they're going to meet in Kuala Lumpur, but I think they started in London, they go to Boston, San Francisco. They meet roughly twice a year all over the world. And I think those meetings, the Microbiota R&D and Business Forums, are seen to be the place where academics and, here's the thing, all these meetings have just sprung up almost overnight. If you look at, just Google microbiome or microbiota meetings, there must be a dozen every year, domestically, internationally, and so on. So it's not a surprise you haven't heard about them. But they've been around maybe 2 years, 2, 3 years, something like that. But they seem to be the place where industry and academics come together around, as you point out, concrete goals.
Well, again, I think, we said earlier that there are opportunities here that are perhaps not too expensive with regard to banking of samples, collection of samples from either clinical trials or from epidemiologic studies that are going on, from animal studies. Just a comment, and Matt and I were talking at breakfast this morning, but at Colorado State, we have an animal cancer center that focuses on using spontaneous tumors from companion animals as a model for human cancer, because there are a lot of parallels in those. They have the opportunity to bank samples out of that. We have clinical trials going with both lead and secondary compounds from pharma right now on an active basis. The IOM has just endorsed this as an approach to really explore in terms of an animal model for cancer. So, these are areas that are opening up, and let's take advantage of them and collect some of that sample and begin to look at this. These are animals that live with us, and all the things we heard about in terms of what influence our microbiota probably influence theirs as well. It'd be interesting to see the comparisons. John?
John Vandenberg, EPA, and I'm a liaison to the committee. To the fourth question here, what could institutions do now to move the field forward? It seems to me that as a representative who does a lot of risk assessments, I can see right now where this information is very valuable. And a couple of the presentations really underscore that. I would say what we saw from the presentation regarding the sort of personalized dieting from Israel was the range of responses of individuals to certain diets. And you could see that there was a highly targeted difference, potentially because their microbiomes were different among those individuals, or as individual groups, there were differences among them. And I would contrast that to Stephanie Shore's presentation, where there was extremely tight data for ozone exposures and responses there. Those animals were developed sort of as a set of very common microbiome in those animals. And what we see is the tightness there versus the great diversity in the human population. So what does that mean from a risk assessor is we oftentimes are working with very messy data. It's data that has a lot of noise in it. And the characteristics of that is we try to understand both the average as well as that range. And Joel sort of spoke to this as well in terms of trying to characterize the exposure response when you've got messy data is messy. We use models to do that. However, what we're trying to do is characterize the range of variability in the individuals in those populations exposed to different groups and understand how we can extend that or kind of estimate what it might be in the human population. So it strikes me that as we look at the data from Stephanie Shore, we've got sort of a unitary type of microbiome, that we've got tight data. We look at the humans, we have very variable data. That's informative right there in the risk assessment to help us understand that, in fact, in most of the animal studies, most of the human studies that we're getting in, we don't have any characterization of the microbiome. And so it's one of the factors that we can kind of think about in terms of reflecting upon why the data are as they are, is that we don't have all the factors that influence, in a sense, the difference between environmental exposure and perhaps blood concentration, because the dose is being affected. That's one potential factor. And I think this is where Lita pointed, I think it was Ellen Silvergeld's model that you put up that showed the microbiome as a step between environmental exposure and essentially dose. So what we're not saying is that we're not really seeing that full characterization of dose potentially, which would influence downstream what the target tissue response might be. So it is useful qualitatively now. When it comes to using it quantitatively, that's down the road someplace. But nonetheless, there is value in the near term in terms of understanding the factors that may influence our characterization of risk. Just a comment.
So, John, I think that the human-animal contrast in terms of the presentations is a really good one. I think it also applies, though, as we think about the way we do our animal experiments, because, in fact, the data coming out of the animal experiments in different laboratories from different sources of animals and different housing situations and so on, even under good laboratory practice as we do it now, provides messy data from this perspective.
Yeah, I would just again reinforce that I thought Stephanie Shore's data was just remarkable. How tight that was, in contrast to much of the other data that we're looking at. It was just really very striking to me.
Yeah.
Thank you.
Thanks.
I have a question of Matt with respect to your conversations with your corporate colleagues, corporate connections. And it has to do with the concept of investing in this kind of examination. And have any of them been talking about anything other than we're waiting to see you tell us what to do? I mean, are most of them kind of in that frame of mind? Because I want to suggest an alternative conversation.
Why don't you suggest your alternative, and then I'll
All right. Research is commoditized. Okay, that's the other piece of this thing, of course. Big Pharma doesn't do all of its own research. A lot of preclinical stuff. Very little preclinical stuff. But here's a suggestion that for the price of one corporate lawyer, they could hire a microbiologist, a molecular biologist, and a bioinformatician. And they could outsource all necessary sequencing for clinical trial for drug X for an entire year to collect microbiome, skin, whatever, sampling. Is that an investment, do you think, that they're ready to consider? Because I think that's important not to have them saying, okay, but we can't do it till you tell us what we should do.
Right. So one of the things I learned quickly in talking to pharma was that the worst way to advance the conversation was to tell them what to do.
No, no,
So
yeah.
for me to walk
this is
in there and
about
say,
asking. Right.
you know,
Yeah.
you all drive much nicer cars than I do, and so why don't you eliminate a couple of people that are driving BMWs and then you know, apply that money to me? I've thought it, but I haven't said it. So I think, I mean, I think they are driven by the market, as they should be.
Oh, I agree.
And so they are looking certainly to companies like mine and to basic researchers as the engines that drive their innovation. And they're very much driven by what the size of the market will be for these indications. I think another thing that they're mindful of is the potential regulatory hurdles that might be put in place. And I see that Carl's still here, so I'll just kind of speak to that a little bit with respect to FDA. They don't know what that regulatory landscape looks like. I'll hazard to say that I think they probably would like something that we're doing a lot, because we're doing essentially drug discovery and development as it has always been done.
Oh, yeah.
And so they can see what that looks like. But I still suspect, and this is a suspicion on my part, and so I've never had anybody say this to me, but I suspect that there are bright people in pharma who are thinking, well, we probably should be banking all the fecal material in clinical trials. But if we do that, we open ourselves up to the question or the request on the part of a safety assessment, maybe in FDA or elsewhere, that, well, if you have all those samples, you should examine all those samples. And there is an opportunity to take first to market and turn it immediately into first to failure. And so I really want to, and I know FDA is mindful of this, because I've actually been at a conference at FDA that have talked about this. But I think there's a point where if you're pharma and you bank all that stuff, there's no plausible deniability at that point to say, well, we can't look at that. We didn't collect it. You did collect it. So doing even 16S on all those samples is prohibitive, much less kind of a metagenomic approach. I would argue that a qPCR approach could be quite useful, where for a targeted set of genes, you could very quickly and accurately determine what changes might be in place. But I talked on the panel yesterday about my inability to sleep and my ability sometimes to sleep. And I'll just go back to that same comment that we've approved drugs for decades. We will continue to approve drugs. They're safe, they're effective, they're idiopathic. We hope to be able to describe why some of them are idiopathic in a way that's not a GWAS, but maybe using the microbiome as a discovery platform there. But I do think this will go forward. I am enthusiastic about it, and I think it will walk forward in the way that it's supposed to, which is carefully and with some stub toes, and then eventually it will break through.
Thanks, Matt. I want to sort of begin to wind us down now in terms of the discussion. I'm going to ask the panel just to make a final comment on your takeaway from the discussion that you've heard this morning in terms of what seems to be some of the more important things that we would want to do. But before I do that, we did have one question from the web, and it was about a comment on the impact of the microbiome, especially the gut microbiome, on vaccine response, and whether we know of either results or experiments that are going on at this point. So anybody want to jump in on that?
I don't know of any particular specific implications but
So Ann, say your name.
Ann Summers, sorry. The largest immune organ in the body is the GI tract. 80% of the immune function is mounted in the GI tract, and vaccine manufacturers have been going towards oral vaccines because of their efficacy and apparently lower side effects and so forth. So I think that it's very likely that, that the focus is that, yes, that's where you're going to effectively get immunity and also perhaps mess it up. But I think that it's certainly a worthy area and something that we should foster investigating.
Margaret?
Well, I don't know the totality of the research that's going on, but I know an emerging area in environmental epidemiology is looking at environmental exposures and their impact on childhood vaccine response. There's a nice JAMA paper with Philippe Grandjean on PFAS, and we're also looking at that in our cohort in the developing immune system and vaccine response in children in relation to environmental exposures, and as we discussed before, the microbiome as a mediator and as an immune contributor.
So that's a good example where those pieces have come together in terms of the studies that are underway.
We don't have the data yet.
Right.
But.
Right. So let's just run down the line, and Yvonne, I'm going to start with you, and then we'll just take a few minutes here just to give your final thoughts.
Yeah, thank you, Bill. This has been a very informative meeting, I think, and the session as well. I want to go back to the beautiful title that we have for this particular workshop, which is Environment and Health. What's the human microbiome have to do with it? And I think that frames my comment, which is, again, my call is to the large consortia that are examining the state of the microbiome and the effects that microbiome has on health, and to inform the environmental research community about the most important factors that are either modulators or modifiers of the effects. And I think that in itself will be very informative for us to ask the next question, which is what is the relationship of those factors with exposure and health effects? So that particular information is crucial for us not to, go shooting in the dark, because our dimensions are yet larger.
Thanks. Margaret?
I guess I would agree and build upon that it is important that we have a methodologic framework and refine that as we go forward and as data emerge and as we make triumphs in the field and discoveries, but also our mistakes and what things could be done and where some of the biases might be introduced in our analyses. And again, moving forward to develop the methodologies, statistical, bioinformatical, as well as epidemiologic, analytical, et cetera, that we need to move the field forward. But it is an exciting time. And, yeah, it's great.
So I think comes with that a call really for a lot of communication on how the field is moving and advancing and letting people learn from the kind of work that's going on. Yeah, great. Matt?
So I would just encourage us to continue to be molecular. There's always a molecular cause to everything. We are not particle physicists. We don't get to point to things like entanglement, which is spooky action at a distance, faster than the speed of light. If there is an effect, there is a chemical reason for that effect. And so we have to have that arc include all the way down to atoms, through animals, and into humans. And so I encourage us to continue to do that.
And Lita?
My take home from this day and a half workshop was how daunting this community, what you're faced with, because you're talking about shifting baselines, variability, not only in your human subjects, but also in your toxins and pollutants that you're studying and so on. And of course, the microbiome is also changing. So that's a really daunting task. However, this community doesn't have to feel alone with it, because you can see by the data I showed that there's a burgeoning microbiome research community. They all embrace open access data and tool sharing through SourceForce and so on. So I think the community that you see rising up that's studying microbiome, there are certain principles that they embrace that will help this community. Open data sharing and computational tool sharing and model sharing and so on. So I want to support your comment, Bill, that I think communication between these communities, the questions that have arisen in the last day and a half, you're not the only ones asking those questions. So let's, find the meetings or create more workshops or do something so there is better crosstalk between the various communities that are asking basically the same questions.
So just a final comment from the moderator. I think from my perspective, as most of you know, I tend to be a glass half full rather than glass half empty type of a person. I look at the parallels between how the field of toxicology and risk assessment is moving forward and the way that we can actually move this particular aspect of toxicology and risk assessment mediated or modulated or influenced by the microbiome. I think that we began a long time ago thinking about chemicals as though they had individual actions, and that we could characterize those actions, and those were separate from biology and physiology and so on. And I think what we recognize now and what our field has moved into is this idea of understanding the commonalities in terms of how biology is affected by or affects chemicals that we interact with. And so adverse outcome pathways, the 21st century toxicology that we talk about provides us with the kind of tools that we're talking about here to begin to address some of these questions if we get beyond simply the genetics and really look at the functional characteristics of these modulators that we're talking about. And I agree with many of the comments. I think Joel said it best, but we have the tools to be able to begin this and to really start to put some perspective around potential impacts that will affect environmental health vis-a-vis the microbiome. And so I think that's encouraging to me and something that will allow us to go on. Hopefully it won't be 5 years until we hold our next workshop, but I would hope that in a few years we'll be able to come back with even more examples of impacts on environmental health that have been mediated by the microbiome. I'm certainly encouraged by the things that I heard here. We're going to take about a 15-minute break right now, and then we're going to come back and Helmut Zarbel will be essentially giving a workshop summary. Those are always a highlight of these workshops, so I encourage you to stay around. Stay around, and we'll be back with you in about 15 minutes. Thanks to our panelists for a very good discussion.